Stanford University.
Okay, let's see if we can make this. At some point when everyone's done, just wave at me and we'll take a few minute break. But what we have, just sort of to review, we started the semester with a Big Bang. And that was Seth Shostak's lecture, and then we started to discuss where you get the elements in the universe, and the elements then forming the building blocks of life. We had Jeff Marcy come in and discuss how you might find planets elsewhere that would have potentially life on them, and so that's very exciting. And from there, we started in on the origin of life, which unfortunately got a little bit reversed, discussion of what is life, and then into sort of this great moments in evolution. And the more I think about it, I think next year I'll just entitle the slide Great Moments in Evolution and pick the sort of the fun ones. And obviously the origin of life is there. The question of when life actually arose, the possibility now that it arose within 100, 200, 300 million years after the Earth formed. The Earth formed 4.6 billion years ago. So in a geological sense, that would be extremely quick, but we do see signs of liquid water on the fairly early Earth. We then had this bombardment period, and we now know that life potentially could have survived through this bombardment period if it had risen very early. We see presence of fossils, both morphological fossils and chemical fossils, very quickly after the bombardment period is over. And then we discussed some of the, what life might have been like at that point, the different domains of life, oxygenic photosynthesis I didn't really push, but that obviously was one of the great inventions. Sex was one of the great inventions, individuality. There were many marvelous inventions in the last couple of billion years, all the way up to leading to, dinosaurs and NASA and the Space Agency and so on. So that's where we took you through last time. And at the end, I really sort of rushed through trying to make the point that evolution occurs in a physical context. And I know in some ways that sounds very Lamarckian. Maybe I stepped out of the wrong century. A lot of people think that. But in a way, it's very important to go back to that kind of thinking, and I think it doesn't tend to be stressed in biology classes. There tends to be a lot of emphasis on competition among species for scarce resources and so on, more of a Darwinian view. But in fact, life evolves, life lives in a physical and chemical context. And some of the things that we talked about specifically last time quickly is solar luminosity, that the UV regime on the Earth, and in fact the whole solar luminosity has changed because the sun is a star. And as things burn like stars, you go from hydrogen to helium and so on, and it becomes hotter and hotter. So in fact, when life arose on the Earth, the sun was only about 70% as luminous as it is today. So if you went outside, the sun wouldn't look as bright. But we also know that young stars tend to emit more ultraviolet radiation, so maybe there was just as much as today. We know the atmosphere was very different. We think the rotation rate of the Earth was very different. It's slowed down because of a frictional interaction with the moon. This is something that's come up a couple of times during the semester. The atmosphere was different. We know that there must have been some greenhouse gases in the atmosphere to keep the Earth basically in a position where it could have liquid water throughout history. And since the sun was less luminous, there must have been some way to do this. And the current thought is that there was some kind of greenhouse effect going on in the atmosphere. And specifically, there was probably a great deal more CO2 in the atmosphere, possibly orders of magnitude more, probably more methane, which is a very effective greenhouse gas, and various things like that, but primarily CO2 and then secondarily methane to keep the surface where it could actually harbor life. Land areas were very different. I showed some pictures with continental drift, which are very interesting to look at. Obviously, if you've got these big land masses coming together, form Gondwanaland, and then it's breaking up and moving around, and Africa goes here, and South America goes there, and so on, that's going to have enormous effects. Tectonic activity, volcanoes, and so on, all these things are going to affect life. And I really wish we could spend a whole semester on some of those, because it's tremendously interesting to think about this interaction with the physical environment, which leads perfectly into this lecture. What if, instead of just talking about relatively normal processes like having South America and Africa drift halfway around the globe, or snowball Earth or something, what if we actually start to look at environmental factors that really push the extremes for life? What does that mean? Okay, so that's what we're going to discuss today. This slide should look familiar from the first class. Astrobiology has 3 big questions that it tackles. Where do we come from? Are we alone? And where are we going? And I'm going to argue in the next hour or so that extremophiles fit in all 3 of these panels. That to understand the evolution of life, what else might be out there, and what's going to happen in the future, this stuff's important. So what I'm going to try to do today is stick with this outline. We're going to go over a little bit what's an extremophile, why study them, environmental extremes, how do they do it, so on and so forth. This is that Laguna Colorado that you may have seen me standing near during the movie last time. Red, Colorado colored, up in Bolivia, the Altiplano. So let's start with what is an extremophile. Well, this word is, in the astrobiology community, is a little bit like saying cancer in the medical community. Everyone puts the word extremophile on a paper or a talk because then it sounds like you're really cool. But, when someone says, well, I work on extremophiles, they're in a cave. Yes, it seems extreme in colloquial English because living in a cave is just not my idea of a great way to spend the afternoon. But in fact, I believe that there are actually definitions for extremophile. And I will say, in the spirit of full disclosure, I got into a lot of this stuff because I was going to extreme environments to try to find communities that mimic the early Earth. And if you start to go to ones that are more extreme, you drop out things like fish and other sorts of things that we're used to. And so these made good mimics. But I never really thought of myself as someone who worked on life in extreme environments. And then about 10 years ago, I got a semi-mysterious email from Nature magazine one day, which is, as you know, the top science journal, saying that they wanted me to write an article on life in extreme environments and how that might relate to looking for life in the universe. And in a show of uncharacteristic modesty, I wrote back and said, I'm actually not an extremophile person. I'm probably not the best person to write this. And they wrote back and they said, we like the way you think. And so, what are you going to say? So obviously I said yes, not knowing much about this. And so I really started from first principles. So this talk is very much that paper that I assigned for you to read, except the updated full color, version with enrichment. Since then, I seem to give talks on this stuff. I gave one last Wednesday. I mean, this is like, I'm now supposed to be this walking Guinness Book of World Records. And I got a couple of other papers out, some that go more into the mechanisms and so on. So people who are really interested in this, I've got lots that I can give you. So this is just sort of the overall summary on steroids. So anyway, we'll go back to now the main event. What is an extremophile? And there isn't really a great definition. So I went back to first principles. Anyone here take Latin? Greek? Oh, there's one at least. We often have a few in here. That's why I was curious. When you go back to it, extremophile is actually made of 2 words, extremis, which is the Latin, meaning being on the outside, plus the Greek philos, which of course means love. And so you end up with this Greco-Latin hybrid. Now, the person who coined this word was Bob McElroy, who was at NASA Ames working on these sorts of things. The question is, are extremophiles really organisms that live on the edge and love it? And so let's look a little bit more about that. First, what does it mean to be in the extreme? I think there, as I sort of jokingly said a moment ago, a tendency to think, well, extreme is something that would be unpleasant to us. Extreme would be New York in August. Extreme would be New York in January. Not to pick on New York, of course. Chemical extremes, what does this mean? Not enough water, maybe? Maybe biological extremes, too much crowding. But that's all kind of fuzzy and, new agey. That's not really very good. maybe extreme means in the eye of the beholder, I thought. Well, maybe, it's something that's extreme for us. Well, that seems very anthropocentric. So I sort of eliminated that. Maybe there's an evolutionary definition. Maybe extreme means anything that's really distant from what the conditions were for the origin of life. So it's something that's very far from the original conditions. That's kind of dumb. Is there an objective definition? And so I started thinking, well, all life on Earth, and as I've argued before, anywhere probably in the universe will be based on organic carbon. So I would think that an extreme condition would be any condition where it's very difficult, if not impossible, to keep a carbon-based body together. Now, obviously, for example, if you go to the surface of Venus, which is over 1,000 degrees, you're going to turn into a little pile of ash, if you're lucky, probably more CO2 and so on. And so that's got to be in an extreme environment. Now, what about love? Do you have to love this extreme environment? And we tend to be schizophrenic because, in fact, most of the thermophiles that we're going to talk about, organisms that live at very high temperatures, cannot live at anything but those very high temperatures. But in fact, we also talk about organisms that withstand very high levels of radiation, radiophiles maybe, but in fact, they don't love it. They don't wake up in the morning and say, zap me. It's just that they're going to be able to survive it. So this love thing is a little, little funky. And then the other point I'd like to make is, does this have to apply to all stages in the life cycle? Now, it turns out that frogs in temperate regions disappear in the winter. Where do they go? They go under lakes and so on. Some of them can actually freeze solid. What happens in the spring? They defrost and they go hippity hop. Now, if you take a frog from a pond in August, stick them in your freezer for a week, and then put them out on the kitchen table and say, hippity hop, froggy, you're not going to have much luck. You're going to have a little mush of defrosted dead frog there. Very sad, very sad. I can see tears welling up in some people's eyes already. It's not going to work. In fact, the reason the frog's able to handle this normally is that as it's getting colder, they're building up antifreeze in their tissues so that they're ready to become an extremophile. So it's not at every stage in their life cycle. Deinococcus radiodurans, who you'll hear about a couple of times, is the champion right now, the champion bacterium in withstanding levels of radiation, very, very high levels of radiation that I would not recommend to your worst enemy. The way they do it is their DNA actually fragments like anyone else's. The difference is, like a jigsaw puzzle, they're able to put it back together. It's absolutely remarkable. But it turns out the Deinococcus radiodurans won't do it if you don't give it enough, I believe it's the manganese. Manganese. If you give them magnesium instead, they won't be able to put themselves back together. So it's not that there's something totally special about the organism in any situation, but it's under certain circumstances if certain elements are available. Okay, so why would you even want to do this? Obviously, there are a whole lot of reasons besides astrobiology, food preservation, basic research. There are people who use extremophiles simply because then they can get maybe whatever solution they're studying into solution because these organisms live at low pH. Biological warfare, we could think about that with aerobiology. Biotechnology potential, this has been a very, very big thing. Now, I'm a federal civil servant, so I've taken a vow of poverty, but many of you may want to actually grow up and make money, and there's a huge amount of money in this stuff, and I'll mention that at the very end of the lecture as well. There's clearly a future use in space. Biodiversity on the Earth, that's some of our interests. Understanding how organisms can survive in very extreme environments, and the limits for life in the universe. And obviously, it's these last 4 bits that give us the excuse to talk about it today. Is this a new idea, the idea of looking at extremophiles to try to get an idea of what the extent of life is in the universe? No, it turns out that Richard Proctor, who was a British scientist and popularizer of science at the end of the 19th century, talked about this a lot, but particularly there was a book that he published in 1870 entitled Other Worlds Than Ours, which has all the sort of, florid 19th century language, but it's basically saying the same thing that we're going to be talking about today, that if you look over the Earth, if you range over the Earth from the Arctic regions to the torrid zone, we find that none of the peculiarities which mark the several regions of our globe suffice to banish life from its surface. So in other words, it doesn't matter how extreme it is, we seem to see life all over the place. And then he spends the rest of the book tying in these different life forms with his hypothetical Mercury life form and a Venus life form and so on, in places that we don't actually look for life today. But this is an old approach, and it's an intriguing one. So which of these taxa that we've been discussing the last few days, taxa just being the plural of taxon, so in other words, which groups might have extremophiles? Well, this tree should look familiar. The bacteria, the archaea, and the eukarya, and every one of them has organisms that I would consider an extremophile. So I want to disabuse you of another common misconception about extremophiles, and that is that they're only found in the archaea. I mentioned last time that the archaea have a certain number, but there are extremophiles all throughout the three domains of life. Okay, so what are some of these environmental extremes? Well, first, let's go back to this whole question of why you're extreme. You're extreme because carbon needs to stay intact if you're going to be a carbon-based life form. And I think I went through these arguments before, but just in case I've missed it, the reason we keep focusing on carbon, coming back to carbon rather than something like nitrogen or silicon, is it is the fourth most common element in the universe. There's a lot of it out there. I realize it's insignificant compared to the amount of hydrogen, but you can't make life out of hydrogen. And so you start to focus on something that's reasonable, and carbon, there's a relatively large amount of it out there. And it's capable of forming these really complex molecules. We saw methane earlier today. That's the simplest organic compound you can come up with, CH4. But it can also form these very long chain fats, amino acids, of course DNA, RNA, and on and on and on and on. And so there's a very complex chemistry of carbon, and we know that that's possible. And beyond that, the sort of stuff that Lou Amendola was talking about, to me, is just really drives the point home that when you look into interstellar space, what you find is not a complex nitrogen chemistry or silicon, or, I hate to, if there are nitrogen silicon fans here, I'm sorry if I'm offending you today, but the fact is you see this carbon chemistry. And as Lou mentioned, in addition to seeing it out there, when you simulate those conditions in the lab, you produce a carbon chemistry. So it seems that this really is the language of the universe, and this is really what's available for life, not just on planet Earth, but anywhere you might go. So we'll get back to the idea that extreme means it's very difficult to keep your body intact if you're a carbon-based life form. And so there are a whole lot of categories of extremophiles that we'll cover today, and this is basically the full color version of one of the tables in that paper, so don't even try to write any of this down. But this is to sort of give you an idea where we're going. We're going to look at temperature extremes. I think I then went on to pH, radiation, pressure, desiccation, salinity, oxygen tension, chemical extremes, vacuum, and so on. So there are a whole lot of these physical and chemical extremes, and I'm sure if you look at that long enough and you think hard enough, you probably can come up with things that aren't even on there. What I find kind of interesting is a few years ago I was asked to give a talk on the history of extremophile research, which is the reverse of doing a real science talk, because you try to find the very first article that was published on it rather than the most recent article. And the more I started thinking about it, the more I started realizing that really all the early work was inadvertently done on extremophiles because people have been interested in food preservation for literally thousands of years. And the way that we're able to preserve food, of course, is by keeping other organisms from growing on it. And this absolutely tends to parallels. So I put together this kind of fun chart, which isn't published anywhere else, but there's the temperature, radiation, pressure, and so on again, and the method that we use for food preservation. So you won't find this in print anywhere else, but you'll have the slides to look at. And of course, it's because high temperature and low temperature retard growth of other organisms. High radiation retards growth of organisms. High pressure, desiccation, of course, is one of the oldest ones, this drying and curing. Salting organisms, salting things like fish and meat and so on. Osmotic pressure, so in other words, something like jam or jellies where you've got a very high amount of sugar. pH, think of sauerkraut, for example, as a way of preserving cabbage with a very low pH. Oxygen tension, taking out the oxygen that's there. Chemical extremes, for example, preserving fruit and ethanol. Other sorts of preservatives, CO2 enriched atmosphere. Smoking meat, that also prevents organisms from growing on it. So it's kind of fun, but if you think about it, we've all sort of, we humans have figured out a lot of these extremes by looking at food preservation for, they say, literally thousands of years. So now let's start to go into some of these extreme parameters in a little more depth. And temperature is the easiest one to explain. You can explain this one to a 2-year-old. Ouch, hot, and they've got temperature pretty much figured out. So there's temperature for you. Like that? Don't you love Keynote? Why does temperature matter? Well, there are a couple of reasons, and I'm going to mention some of the little more subtle regions first. First of all, the solubility of gases in a liquid is very much temperature dependent. If you don't believe me, take 2 cans of soda, put 1 can in the refrigerator, and put 1 in some hot water, and then open them up and tell me which one is going to go. Which one is it? I don't know. Well, since you seem somewhat unsure, I recommend you go home and try that experiment and come back next Tuesday and tell me. And I think you'll find that the one that's been hot has a much lower solubility of the gas, which is a nice way of putting that if you're wearing a white shirt and you've just opened a hot can of Pepsi, you may regret this. So if you're, say, photosynthetic and you're aquatic, like an alga, and it's getting hot, the solubility of carbon dioxide in the water's gone down. If you're, say, a fish and you're in water and it's getting pretty hot, the solubility of oxygen has gone way down. And my understanding is that's what creates the upper temperature limit for things like fish. Now, being somewhat sadistic and being a microbiologist, I've always wondered what would happen if you start to raise the temperature a little bit in a fish tank but bubble a little bit more oxygen through it. Now, obviously, you can't poach the fish. There are going to be issues there. But you might be able to buy yourself a few degrees. So anyone who wants to go try that, I strongly encourage you to come back and report to us. Second point, enzymes have optimal temperatures. So if you've taken biochemistry, you remember all these curves where you see something, an enzyme maybe not being functional at all at maybe 5 or 10 degrees and then has a peak functionality at 20, 25 degrees and then goes back down. So you want to sort of optimize these enzymes. But even more than that, you have various biomolecules that are very temperature sensitive. And I think the best way to describe that is think about frying an egg. Now you start with this egg that basically has a transparent white and a relatively transparent yolk. You fry it, what happens? The white turns opaque, the yellow turns opaque. The white turns opaque, why? You know why? Because you're denaturing the proteins in it, the albumin. When you denature something, what's happening is you're unfolding it. So you've taken this protein that poor bird has carefully constructed in its optimal form for whatever it's evolved to do, and you've now unwound it, pulled it, torqued it around, and that's all she wrote. If one of you knows how to unboil it or unfry it, then you need to see me at class because we need to file for a patent. I don't know any, I always offer, but no one yet has had the nerve to tell me that they have a way to unboil an egg or unfry it. But it's a great idea, isn't it? The reason it's so difficult is you'd have to figure out how to untwist all these things that have been denatured and put them back to where they were. Now, before you even put the egg in the pan, you've probably put something like butter in there, and you've melted it. So what you're doing is melting these lipids. You're turning them into more of a liquid. Now remember, your membranes on your cells are also basically lipids. Well, they are lipids with some proteins in them. So if it gets too hot, you're basically melting cell membranes. conversely, if you stick that butter in the freezer and then you take it out, you can hit it over the counter like that frozen frog, and it's going to be hard to crack. So if it's cold enough, your cell membranes are going to lose a lot of their fluidity. They're going to get very stiff, and that's going to be extremely awkward as well. There are ways to get around this a little bit. You can buy a few degrees, and we'll talk about them later. But I'm going into the temperature in much more detail so you can get an idea that there are a lot of subtleties involved with why these physical and chemical extremes are so difficult for us to live in. Finally, if you're in very cold temperatures so that there's ice forming, try to imagine for a moment that you're shrunk down to the size of maybe the nucleus of a cell. You've got ice crystals forming. They're going to look to you sort of like swords cutting right through. And now picture that your cell is like a water balloon. You've got these swords being shoved right through these water balloons. What's going to happen? The insides are going to leak out, and that's all she wrote. And that's why your frog that you shoved in the freezer in August isn't going to do so well when you take them out, because a lot of the contents of the cells are now going to be leaking all over the place. So he's dead. He's no longer a live frog. So that just drives home the point that we all have our optimal temperature. So now that you've thought a little bit and maybe you're a little more sophisticated about why temperature matters, let me just give you a couple of examples. In many cases, I have a lot of writing on these slides, and it's so that you don't need to worry too much about taking notes. You can go back and get this information. I don't expect you to read it all right now necessarily. But I believe I mentioned Tom Brock last week, and now I'm getting confused with the lecture I gave Wednesday and the one that I gave to you guys on Tuesday and Thursday. But in case I didn't, Tom Brock was a young professor at Indiana University. When someone encouraged him to stop in Yellowstone and look at some of the ponds there, he was doing more medical research at the time. And he did, and this is a perfect example of why sometimes you shouldn't listen to your other people who say, well, it's impossible to live in high temperatures. As you know, there are a lot of hot springs in Yellowstone, and he started looking in some of these and seeing things that looked like they were probably alive, and lo and behold, they were, and ended up having a huge research program in Yellowstone over the course of 20 years or so with armies of graduate students, many publications and so on. And that's some of the history right there of, from his autobiography. And what happened was he then published a paper that I believe was in Science in the early 70s talking about life at very high temperature. And once I started interviewing people who have worked on extremophiles, high salt, and so on, many of them were inspired by this work of Tom Brock's in Yellowstone. Very cool guy. He then ended up in Wisconsin and is now retired for the most part, but occasionally puts out an article or two. NSF also started then funding research down at the very deep sea and discovered these communities at the hydrothermal vents. So again, very, very high temperatures, basically similar to what Brock was studying, except Brock's hot springs were at the surface, of course, and Alvin was able to get way down, hundreds, thousands of meters beneath the surface to some of these hot springs. Very, very cool stuff. So what are these limits for life? Well, if you look at the low end, my understanding is that the Himalayan midge is active at -17. I have no personal experience with the Himalayan midge. I am taking the published literature at face value. So it may turn out that, there was some Himalayan midge chauvinist, and in fact they can't go below -10 or something like that. I don't know, but I'm just going to leave it there. There are reports of some microbes being active at about -20. When I actually looked at the data in the papers, it was unclear to me quite how they interpreted that. But there are some that are, this is now tens of degrees below freezing. If you start to go near freezing, like at about -5, there are lots of organisms that can do that. And basically what they're doing is making some antifreeze, just like you put in your car if you live in cold areas. So they're fish, fungi, algae, protozoa, bacteria, and so on that can buy a few degrees. If you're living in salt water, you are also buying a few degrees by the water-stained liquid at a few degrees below zero. And then so these are the so-called psychrophiles, the ones that like very cold temperatures. Then you get into the mesophiles. organisms like us, plants, mosses, fungi, algae, most of the animals that you know and so on are all mesophiles, so sort of middle range there. And then you start to get into thermophiles, and these are really sort of starting at 60 or so. Different people have different definitions, use whatever definition I have in the papers. And now you start to have other organisms drop out. As I said, animals are around 65 or so. If you look at that book that I recommend on the syllabus and at the end of this talk, they talk a little bit more about animals at some of these extremes and humans being able to survive very hot temperatures for very brief periods of time. I do not have malpractice insurance. I'm not going to recommend it, but I do recommend reading the book. But as you start to get warmer and warmer, plants drop out too, and that's because chlorophyll is going to break down at about 72 or 73 degrees centigrade. This is hot, but it's nowhere near boiling, of course, which is at 100. So at some point you drop out all eukaryotes, and now you're in the realm of bacteria and archaea, and at some point even the bacteria drop out, and you're ending up basically with these very high temperature archaea. And this is where this rumor got started that the only extremophiles were high temperature archaea. Not true, but I readily admit, even though I'm a eukaryote chauvinist, as you know, that the archaea are very definitely the high temperature limits. Now occasionally you'll see hype about organisms being found at much higher temperatures. I have 121 there because I think that that's the highest temperature that there is good evidence for. But that doesn't mean that tomorrow in the paper or next week or next year or in 10 years, someone won't publish an equally good paper saying that they found something that lives at 130. Now, I don't know how far up you can go, because when you're talking about temperatures like that, and particularly if you're an archaean, you have to do an awful lot of jerry-rigging of your body. Is normally the DNA is going to denature, your protein's going to denature, your lipids are going to go bleh. I mean, you really are not going to do very well at those temperatures. And so for these organisms to be able to pull this off has required a whole lot of engineering. So this is something that once they've made that commitment, there they are. Now, I put the Pompeii worm over there because I think that's an intriguing little example. This is an animal that lives at higher temperatures. It lives by hydrothermal vents. How does it do this? How does it beat the odds? Well, I get different answers depending upon the age of the group, but I'm going to assume, without embarrassing you, that everyone here has seen at least one Harry Potter movie or read at least one Harry Potter book. Is there anyone who is, you don't want to admit it, so we won't even, just go do that. That's another one of your assignments this week. So when I say Hagrid, all of you know exactly what I'm talking about, the giant. Now, my understanding from stealing my children's Discover magazines is that his costume was very heavy and very hot. And so what they did is they ran tubes of cold water underneath that coat to keep the actor cool. That's exactly what the Pompeii worm does. They live right near the edge of these hydrothermal vents and can stick a tube out where it's very cold on the ocean floor and run cold water between their body and this casing that they have and keep themselves cool. Now, think about that. You got that situation, and then I just told you, well, nothing much can survive below zero, but another quick movie quiz. How many people here have seen The March of the Penguins? Or any movie? Or anyone ever not heard of a penguin? Everyone's heard of penguins. Penguins live in absolutely miserably cold conditions. The ones that are on vacation in Tierra del Fuego or something. But the ones in the Antarctic, it's miserable down there. They're freezing. I mean, obviously, it's much lower than -5. How do they do it? Doesn't take a rocket scientist. They've got feathers, and they've got fat, and, all that sort of stuff. And what's fascinating is or at least I used to say this at professional seminars, my colleagues, and invariably someone would say, oh, but that's cheating. They've got feathers. It's like, get over it. There is no rule book that says, excuse me, chapter 2, section 3, if you have feathers, you don't count as an extremophile. These are adaptations. This is real life. The Pompeii worm counts. Penguins count. Okay? So when you start to think about it, you realize that life has beaten those odds between 0 and 100. They are not so hard and fast. Okay? So I'm really belaboring the temperature bit. We won't spend nearly as much time on the other extremes, but I'm doing this to show you that we could do the same thing on almost every one of these any one of these environmental extremes. So I just wanted to show you a picture or two so that you can get a feel for what some of these high temperature communities look like, and then we'll go back to that towards the end of the lecture. We'll look at some of these other communities. This is Octopus Springs in Yellowstone National Park. How many people here have been to Yellowstone? Ah, we should all go. It's great. Every year people say they want a field trip. Someday we'll do, that'll be fun. We'll go to Yellowstone spring break, which will be absolutely miserable time to go, but it'll be empty. So this is Octopus Springs. It is not too far from Old Faithful. It's a little bit north of it. We'll be showing other pictures later that are a great deal farther north. It's about pH 8, so nothing too unusual there. And because the altitude, the source where the water's boiling, is about 95 degrees rather than 100 degrees, yet there are high temperature archaea, hyperthermophiles, that live in there. Now, as the water, this is, by the way, octopus, because if you sort of lean your head over to the left, you'll see it looks a little bit like an octopus on its side. If you start to look at that outflow towards the bottom, you see a pinkish looking leg, and what I have marked there is Thermocrinis ruber. There are a couple of other organisms in there too. Now, that's at 83 degrees. 83 degrees only sounds cool if you've been talking about 100 or 121. 83 degrees is, well, the technical term for it is damn hot. And I know this because I accidentally stepped in that once after a very long day, and it's only about this deep, and you have never seen anyone take off a boot so fast in your life. It is damn hot, okay? Going from damn hot to extremely hot, you see that outflow channel, and what you notice immediately where it says 75 degrees is that the inside is not colored, it's clear, but the edges are green. That's right around that temperature where chlorophyll breaks down. So when it's too hot in the middle, there's nothing that's photosynthetic. As the water starts to cool in the edges, as soon as chlorophyll can stay intact, you see photosynthetic organisms. If you go down a few more degrees in one of those upper channels, you see a microbial mat at about 65 degrees. This is very much like microbial jelly, or jello, I guess is probably even a better description. It's about this thick, and on top you see these photosynthetic cyanobacteria, Synechococcus, a few other things, and then you see bits of red and so on, Chloroflexus and other sorts of bacteria in there. Now, 65 degrees in this grand technical scale is not damn hot, and it's not extremely hot, but it's still pretty darn hot. Okay, so that will not be on the final, but just so that you have a good conception. I can stick my hand in there to pull out a sample to do experiments, but I don't leave my hand in there. I grab and come back out very quickly. Okay, so yet there's obviously a very rich, very well-developed microbial community there. Okay. So let's move on to pH, because we've spent lots and lots of time on temperature now. pH, if you remember from your chem classes at some point, has to do with the concentration of hydrogen ions, or if your textbook said hydronium ions. So basically it's a concentration scale. If you want to get a little more technical, it's the inverse of the log of these hydrogen ions. What does that mean? If you're starting to talk about a log scale, 0 doesn't make any sense anymore. So what 0 is on a pH scale is not the absolute minimum. It's when you have a concentration of 1 molar hydrogen ions. When you're starting to get to vanishingly small, you're up at pH 14. So remember, this is a log scale. So pH 1 has 10 times as more hydrogen ions as pH 2, and so on and so forth. This is not a straight scale, it's like the Richter scale. So, as I said, you immediately notice that what you were taught in chemistry, that this goes from 0 to 14, is not literally true, that you can go below 0 and you could go above 14. We live in a world that's in the middle. We call it neutral at about pH 7. The cytoplasm of our cells is about pH 7, plus or minus. What's the pH of the ocean? Anyone know? Used to be 8.2 when I was young. I think it is hovering closer to 8 on an average right now. Sadly, by the time you're all elderly faculty or whatever you're going to do, you probably will be talking about pH 7.8 or something. And this may sound like a trivial amount to the lay public, but keep remembering that this is a log scale. And so there's a big difference between 8.2 and 7.8. It's going to be very sad for some of these organisms. Okay, so this is a huge scale. Down here you're talking about battery acid. Up there you're talking about bleach. Nasty scale, so where do the organisms come in? Well, we'll cut to the chase. The highest pH ones are up around 11.5. There are reports of organisms from lakes at 12.5. It is nearly impossible to keep a culture medium at 12.5 in the lab. So the only reason we're saying 12.5 is that we see natural lakes up there. Most of these are in the Rift Valley in Kenya and in Tanzania, and I'll show you a few more pictures because we've been doing field work there the last 2 years. Down below there's just a little panel from a corner of Lake Bogoria, and that corner is at about pH 11 point something. So this is a lake in Kenya in this Rift Valley where it's been drying up. Yeah.
Does it affect the animals?
Well, there are only a couple of animals. You'll see flamingos, and you notice that I show an inordinate number of pictures with flamingos. The reason is flamingos are adapted for really extreme environments. So when you're at very high salt or very high pH or whatever, you tend to see flamingos. The other animal that you see in there, well, you see a colleague of mine, a graduate student, and my son. And so these were all animals that were either wanted to or were forced into this. No, I'm kidding. You do see some brine shrimp in there. You see some cyanobacteria. You see arthrospiron. You'll see a better picture a little bit later in the lecture. But you notice the water does look kind of bluish, greenish, a little bit darker than your shirt. So it's, and maybe a little bit greener, but that kind of slightly off green. So not like Joshua's shirt, but more your sort of, I guess I should have worn a cyanobacterial shirt myself today. So that's why you see the greenish. So there's a depauperate community, but it's not impossible, and you notice that these are organisms from all over the phylogenetic tree. Yeah?
How does a lake stay at that pH naturally for so long?
Well, it's going to keep going up because what's happening is that these were huge seas that were trapped, and so there's been evaporation over the centuries, okay? And that's why you see it there. So that's all very interesting, but now let's go to the other extreme. And this is where it gets to be very challenging for an organism because you're trying to keep your cytoplasm at about pH 7, and yet you're just surrounded by hydrogen ions, and so you got to keep them out. And so now you've started really dropping organisms out. You start losing things like fish at about pH 4. And so you're down, there are a few flies that maybe can make it to 2. There are a bunch of organisms that can make it down to pH between 0 and 1. Here is Congress Springs. This is where Tom Brock originally found Sulfolobus. It was very dried that year. This is also in Yellowstone. The only way really to describe Congress Springs is boiling battery acid. Yet this is where Sulfolobus lives. Sulfolobus is not very happy any other place unless it's in boiling battery acid. So now you're talking about very high temperature and extremely low pH. What does this mean? If we were to go to Venus-like conditions where you see boiling battery acid, you wouldn't say, no life here, pack it up, let's go have lunch. You would say, well, you know, when I took astrobiology, I found out that there were organisms that can survive in boiling battery acid. So maybe this is not an incredibly stupid place to look for life. We know on Earth it's at least possible. There I am playing at about pH 2.5. This is also in Yellowstone. And I need to say this because this is being filmed. You cannot go to Yellowstone and just walk into any of these places. Every slide you see of me in places like Yellowstone, and actually this goes for Kenya and Australia and so on, you have to get a permit, and Bolivia as well. So I have permission to do this. Tourists are not supposed to do this sort of thing. Anyway, this is Nymph Creek. I've now said that for the camera. This is Nymph Creek. It's about pH 2.5 to 4. It varies a little because the underwater plumbing changes a little from year to year. But you see there's obviously a very rich microbial community. You can see immediately it's almost a pure culture of red algae. What's wrong? Doesn't she know her basic colors? These are red algae, but they don't have a lot of red pigment covering their green chlorophyll. So they're phylogenetically, they're evolutionarily red algae, but they don't look red. And there's a micrograph that I took of a field sample. So you can see it's almost a pure culture. This is not a cultured sample, this is just a field sample. So it's almost all this organism, Cyanidium caldarium, and actually one or two of its very close relatives. They do very well at low pH and warm temperature. This isn't too hot. As you notice, I'm standing there rather than screeching and running into the woods. It's about 40 to 50 degrees, so it's very pleasantly warm. This is more, getting to the sauna region. And so it's a very pleasant place to work. Now, if you take them into the lab, which I've done, Cyanidium is happy to grow from down there pH 0 to about 7 or 8. It also does fine at room temperature. And so I've come to the conclusion it's just simply a lousy competitor. It cannot outcompete anyone else at more clement conditions, but it happens to be very good at this low pH, warm sort of niche. And when you go to places in the same pH and temperature in, say, New Zealand, you see exactly the same thing. So it's very good at that combination of environments. Oh, we're not doing too badly. So we're going to start going even faster now. Salinity. You probably haven't thought about salinity before, but salt is extremely important. You need the electrical conductivity. You need that for your DNA, your proteins to function, all these enzymatic reactions and so on. You need some solutes. But if you have too much, you've got a problem. Because remember, your internal salt concentration is going to try to match the external. I'm wondering if I put a slide in here that'll really show that. However, there are organisms that can live under very high concentrations of salt. These are called halophiles. And what you see in the top picture is from a salt company in Mexico. Actually, it is the biggest salt company in the world, Exportador de Sal. It's about halfway down the Baja Peninsula. I used to do a lot of work there. I haven't been there in a few years. But it's jointly owned by the Mexican government and Mitsubishi. And it's, listening to the Japanese speaking Spanish is a treat. This is a very dry area. There's an inlet from the sea, and so it's a perfect place to produce salt by evaporation. Cargill Salt Company out in the bay does the same sort of thing, but obviously it's not nearly as warm here and it's much wetter. So Mexico is a much better place to make this salt, and the works are unbelievable. If you've ever seen a snow removal truck, picture something about 5 times that size, and these things are going 24 hours a day, 365 days a year. It's an enormously large operation. The salt is for industrial use, not for eating. But in this picture you see one of their concentration ponds right before the salt really starts to precipitate. And what you notice immediately is it's very red. I could have teased you and said what you notice immediately is it's full of green algae, which it is. The green algae are making a red pigment to protect themselves. And so almost always, if you see a very high salt condition like the Laguna Colorada that you saw in the movie and I showed you a few minutes ago, or the salt ponds in Mexico, or the salt ponds in the bay, you see the red. So conversely, when you see water that looks very red, the Red Sea and so on, you can almost invariably say, it's high salt. Those are halophiles. It's very impressive. Next time you fly into SFO, you just sort of nudge the person next to you and say, I bet that's got a high salt concentration. How do you know? Well, red, because you go to Stanford and you need to know these things. Yeah.
Are tourists not allowed in that area as well?
Yeah, in the salt company you certainly do need a permit. There are parts sort of on the edge. For example, where I got that evaporite down below was at the airport, and you certainly don't need a permit for that. This company's in Guerrero Negro, which is also where there are a lot of whales that calf, and so it tends to be big sightseeing tourist areas some time of the year. So you can go to many areas around there where you can get these dried salts and so on, but the actual ponds are kept off limits to the public. just like the ones at Cargill are. We do some work there with permission and permits, but it's actually locked if you drive over there. Okay. This bottom picture is very interesting. This is a dried salt crust. As I mentioned, we got this near the airport in Guerrero Negro. When I say airport, do not picture SFO or Kennedy or something. I mean, this is a little, local thing. But if you look carefully, you see a greenish band towards the bottom, and then I've got a blow up of a micrograph. There are some cyanobacteria that live in there, some chains. And we've done some experiments on them. They are still metabolically active, even under this very high salt and very dried conditions. You can take them back to the lab, leave them on the bench for years. They're not metabolizing very fast, but they're still there. Now, for various reasons, we've also predicted that these organisms should be pretty resistant to being able to live in space, to vacuum, certainly desiccation. We know they can dry out, and to radiation. And so Rocco Mancinelli, who's going to be coming in about 2 weeks, is going to talk to you about when he's taken some of these experiments from these very evaporites and sent them up on satellites with the European Space Agency and exposed them to space and then brought them back to his lab. So remember that picture from when, for when he comes in. Now, as I mentioned, desiccation and salinity can be tied in together. If something is resistant to very high levels of salt, it tends to be resistant to lack of water, to desiccation. Not everything that's desiccation resistant is also a halophile, though, so it's sort of a one-way thing. Desiccation is obviously something that's going to be extremely important because we use water as a solvent. So you can imagine that as you start to dry up, the concentration of everything in your cells is going to go higher and higher, and this is going to be a problem. That's, by the way, another problem with freezing an organism, is that the pure water is going to freeze first, and everything that's left in the cell is going to get more and more concentrated till you're at the danger zone. So that's going to be another problem. Yet there are organisms that can lose much of their body water for various periods of time. Again, I show that evaporite from Baja. But down below, here's a shot from Bolivia, up at about 15,000 feet. This was actually an Inca ruin, so there were people who lived up there, although I can assure you from personal experience that the breathing is tough. There is not nearly as much oxygen as we have in Palo Alto here. And there is not nearly as much water as we have in Palo Alto, particularly this week. What we had in the last week would do these guys probably for a decade. I mean, they just don't see much. So any plant there needs to be able to deal with desiccation. And the reason that this then ties in with radiation, which is what's coming up next, is if you think about it, you're dried up, you've basically shut down your factory, but you're still being exposed to the sun and every other environmental condition. So you're suffering damage from UV radiation to your DNA, to your proteins, to your lipids, and so on, and you can't do anything about it. When you get that minuscule amount of precipitation, you better rehydrate, fix the damage, and then do whatever you need to do. Photosynthesize, make babies, make seeds, all those sorts of things very quickly before you dry out again. So life is tough for organisms that can undergo desiccation. So we're going to move right on into radiation. I'm sure you're familiar with that entire chart. There'll be a quiz on it. This is your basic cartoon version of the electromagnetic spectrum. Now, where it shows visible light is the very tiny sliver that we live in. So we really, our senses are in a very, very narrow band. And that's why, again, I refer you back to Lou Alamandola's lecture where he was showing these galaxies in the infrared. We do know that there are some organisms like the pit python that can see a little bit into the infrared. That's how they find prey at night. They sense the heat and they go. There are some organisms that can sense into the UV, like some moths and so on. Oddly enough, we're not supposed to be able to sense in the UV, but I go out with professional photographers, and you can tell, even though the visible light's the same, I'm sitting there with the meters when the UV's higher, and they'll say, I don't think these pictures are going to look good. So I personally think that we're able to sense a little bit into the UV, but that's not for tampon. I obviously haven't looked at this picture in a while. You obviously don't need to memorize this entire chart. But anyway, the point is there's visible. We go a little bit into the UV, we go a little bit into the infrared, but not much. Most of these shorter wavelengths, UV, are increasingly dangerous to life. The longer ones are not increasingly dangerous, but they have other interesting things. Why is this radiation in both the ionizing realm, so things like radioactive elements, and in the range of ultraviolet radiation, so dangerous? Well, first of all, we've seen some of this here for many years. So some of it we've had workarounds. Some of this is blocked by the Earth's atmosphere, but some of it is going to be increasingly important as we go out into space. Now, the problem with these forms of radiation, with ultraviolet and with ionizing radiation, is that it really cuts to the quick in natural selection. These are mutagens. Not only are they mutagens, but they're selective agents. So they get you coming and going. They not only affect your DNA, but they affect the rest of your body where you can live too. So what I wanted to do is, this is the less cartoony version of the solar spectrum. Again, there's that little sliver where we live. And here's some spectra that I just took a couple of years ago, just outside my window at Ames. I took the screens off, stuck my hand out with a nice spectroradiometer. And what you see is that the spectrum changes very quickly, even in the course of a half an hour in the afternoon. You probably know that. And you can see the UV there, what's below about 400 nanometers. Now, the reason we're so interested in this is that it's a mutagen. Why is it a mutagen? Well, it's a mutagen, and good. That's what I thought it was going to be. It's because DNA and RNA have a peak absorption at 260 nanometers. So you're into the UVB there. So if you're absorbing radiation at that wavelength, that means that you're likely to break or do other nasty things, which I think I'll be showing you in a moment. Protein has a peak absorption at 280 nanometers. It's not so good to destroy your protein either. Lipids are somewhere out there as well. So you get the idea that all these biomolecules absorb in the UV range. So not only is ultraviolet radiation dangerous for all life on Earth, it's likewise going to be dangerous for any carbon-based life form anywhere. As I mentioned, ionizing radiation ain't so great for you either. Don't stick your hand in a particle accelerator or any of these sorts of things either. What I wanted to do is drive this point home. This is a little data that a former student of mine, Jackie Garguet, did I guess about a decade ago. She did a very simple-minded experiment, not because she was simple-minded, but because I asked her to do this. She took yeast cells, and we put them in a water bath on the roof over at Ames, so just a few miles away, nothing fancy. And what we did was we put some, and when I say we, of course, she was the one who got up early in the morning to do this, some UV transparent plexiglass over it, and that's what says UV transparent, obviously, some UV opaque plexiglass, so it let in the visible light, but it blocked all the UV, and some mylar, which lets in UVB, which is a little bit shorter wavelength than UVA. And what you see is that the growth curves are radically different. So these cells are feeling that effect right away. Now, we only have a few freshmen in the class. I think all of you are aware that March is not the peak of summer around here. This is the kind of effect we saw taking out UV in March. So you can only imagine if we'd repeated these experiments in July what we would have seen. So it's driving home the point that UV is deeply, profoundly felt by everything out there. Now, tied in with radiation, this may sound odd, is oxidative damage. Now, all of us breathe oxygen, and that's a marvelous thing. And there's nothing wrong with oxygen. I don't want to discourage any of you from continuing to breathe oxygen. In fact, I would encourage you to continue. Take a deep breath. So oxygen's fine. And there's actually nothing wrong with water, which is where oxygen ends up if you're using it as a terminal electron acceptor like we are. So nothing wrong with oxygen, nothing wrong with water. The problem is all these reactive oxygen species, as they're picking up electrons or shedding them, as you're doing these transformations. And the worst in there is that hydroxyl radical. And so even though I'm only a little bit doing this to tweak you, I would argue that we're extremophiles too, because we're sitting here breathing 21% oxygen. And that means that we are literally playing with fire, because when you're adding oxygen to compounds, you're burning them. So I would argue that all of us aerobes are extremophiles too. Now, again, I don't discourage you from breathing. In fact, we wouldn't be here because we have brains that require huge amounts of oxygen. Basically, all eukaryotes are aerobic. There are only a few that are probably secondarily anaerobic. It's a much more efficient form of metabolism. It's 18 times more efficient than anaerobic. I'm trying now to make you feel a little bit better now that I told you you're doing something very, very dangerous. Without it, you'd have the oxygenic photosynthesis, you wouldn't have large animals, you would barely get tiny little microbes without aerobic metabolism, but it is dangerous. If there's nothing else you remember from my lectures this semester, life is lazy. I've had students suggest that that be engraved on my tombstone. I think that's a little premature, but nonetheless, this is my principle. Life is lazy. You don't do anything you don't have to do. If you come down in the morning for breakfast and your mother has made you eggs Benedict and a nice, pot of hot chocolate and an espresso and goodness knows what else, you don't rummage through the closet and pour yourself a bowl of Cheerios. You're crazy. She's already spent this energy making something for you that you then can take your extra energy and use for something else. You don't do anything you don't want to, you don't have to do, because you need that energy to produce offspring, because that's the name of the game in evolution. I bet my class has the highest birth rate at the whole university. You don't need to do it immediately. I'm just saying that the name of the game ultimately is you need offspring. And so you don't waste energy doing something you don't need to do. So if there's a clever way around it and still being able to live in an extreme environment, you do that. So, for example, let's go back to radiation for a moment. There are all sorts of ways that you can beat the game. One is you can live under some UV absorbing material, for example, enough water or dead cells. And if you think that's kind of funky, that's basically what we're doing. The top layer of our cells is dead, on our skin. You could live, for example, in a salt crystal, which I've shown attenuates a lot of the ultraviolet radiation, but will let enough photosynthetically active radiation through so that these organisms can photosynthesize. I probably should have, I hadn't expected some of the things in that cartoon slide since I haven't used that one before. But in fact, to remind you, photosynthetically active radiation is between about 400 and 700 nanometers. And this is exactly the same region that we see in, 400 to 700. So then 400 to about 320 is UVA, 320 to about 280 is UVB. And that's the one that's being affected by changes in the ozone layer. That's the one we freak out about in the summer. That's why you wear sunblock, because that's the region, even though we don't see wavelengths at 280 nanometers or 260 on the Earth today, it's so close to that 260, the peak absorption for DNA, it's extremely dangerous. So if there's nothing, I feel stupid on a day where it's dark and rainy to tell you wear sunblock in the summer, but put a note on the front of your notebook, note to self, in the spring wear sunblock, okay? And this is why. Okay, so you're living under UV absorbing material. You can produce UV absorbing pigments, and again, I think everyone in this room has ancestors from a region where you had to produce UV absorbing pigments. We can adjust. People who come from more equatorial regions or very high altitudes have dark skin all the time because they need to block that UV all the time. People who come from more northern regions, Scandinavia and so on, have very fair skin because their problem is not blocking the UV. Their problem is getting enough UV so they can produce folic acid so their babies don't have spina bifida. So we as humans have played this game with pigmentation. Certainly algae do it, lots of organisms do this. And you can see there, in fact, a beautiful green algal mat. Once again, I am not colorblind. The purple is the pigment that they produce as they get older to protect themselves from UV, and the very green are the younger filaments in there that haven't made their UV protection yet. We have all sorts of antioxidants in our cells. We have ways to neutralize these reactive oxygen species, all these radicals that I told you about, that can be produced by UV radiation. Or we can simply repair the damage that's produced. The number that I've heard, although I've heard even higher numbers, is that every day, if you were an extreme, I hate to use the word extreme in this lecture, an extreme couch potato who never got up, you didn't go to the refrigerator, you didn't change the channels, you sat there all day, on average, every one of your cells would get 17 oxidative lesions in your DNA, just from being aerobic. Now, if you start to add things into this, like switching the channels or, getting up and eating some pizza, God forbid you actually go to class or run or something athletic, you're bringing that number up. So obviously we have backup system after backup system to deal with all this damage. And this isn't even adding when you go out into the sun and you get the direct damage from UV and indirect because the UV produces reactive oxygen species in addition to your metabolism. And then the poor plants are photosynthesizing where you can also pick up some more reactive oxygen species. So it's dangerous out there. So there are all sorts of ways to fix this. I didn't, I guess, put that slide in because I thought I would have mentioned it by now. But pH is a beautiful example of how organisms can get around this huge pH scale and still stay supremely lazy. If you're at a very high pH, you avoid needing protons for anything useful because you're not going to be able to find them very easily. However, if you're at a very low pH, you're now in this soup of hydrogen ions. And yet you're trying to keep your cytoplasm at about pH 7. So what you got to think about is that your cell is like a boat. You're having to bail. And so what cells, organisms that live at very low pH do is they make very good proton pumps. So it's sort of like bailing water out of a leaky boat. They're bailing these protons out to keep their innards good. So if you're keeping your innards about the same pH, you don't have to re-engineer anything. All you're having to do basically is have a very good proton pump and maybe a few proteins that span to the external environment that are able to deal with the low pHs, but not much. If you live at extremely high temperature, like one of these archaea that I mentioned earlier, you really have no choice. You're going to have to do an enormous amount of re-engineering of your innards, and from a biochemical point of view. So forget being late, you can't get around it. You're going to have to do work. So some of these things, some of these adaptations require really sort of trivial adaptations, and that's why you see it evolving over and over and over. And some of them are very tricky adaptations, and that's why you only see it evolving maybe once or twice. So let me just show you a few of these extreme environments. You'll notice that many of these have more than one extreme associated with it, like these geysers. I told you some of them are very low pH as well as being hot, ice, polar regions, and so on. So let's look at a couple of pictures. Little advertisement that some of the questions that I look at, which I mentioned the first day, are what was life like on the early Earth? How do organisms respond to their environment and nature, and can organisms go beyond the Earth? Every one of these involves extremophiles too. And so I'm going to show you this because the pictures I'm going to show you are all ones that I've taken during field work. As I mentioned, we used to do a lot of work in Baja. I'd love to go back. Maybe we can get a group to go back there at some point. We've done some work up in Lassen. Show you some pictures from there, the salt in the San Francisco Bay, Yellowstone, Bolivia. You've heard us talk about that before. I had some experiments on a Danish ship, the Galathea 3, and that's what that is. We won't talk about that. Some in the Rift Valleys of Kenya, Australia, New Zealand. So let's do a little travelogue for a few minutes here. This is out in the San Francisco Bay, and this is one of those salt ponds at Cargill. You can see from the red ponds from the satellite image on the east part of the bay, southeast part. Some of it's being decommissioned, some of it's being kept there, but it's great because it's a very local field site. That's a picture of Dana Rogoff, who in the lab is my absolute right-hand man. She's sort of my Jessie, except in the lab. You know, I need two people to keep me coordinated. This is up in Lassen. If you haven't been there, it's not that far. It's a national park that's only a couple of hours north of San Francisco. This was the field trip that we took in July. The day before, we were out at over 100-degree weather in the Warner Valley, and the ranger we were with said, we're going to go snowshoeing tomorrow. What size do you wear? I'm like, haha, very funny. It's 100 degrees out here. And sure enough, there was another place in the park that still had snow, which is very cool. And you'll notice that the snow looks pinkish. And again, it's because of green algae that have a lot of this red pigment, these carotenoids, same pigment that's used by these halophiles out in the bay to mask their green chlorophyll and turn red. There are some snow algae that don't look red, but many of them do look red or pink, and so they're called watermelon snow. They also smell a little bit like watermelon, some of them. Here's another very cold environment. These are the lakes in the dry valleys, the permanently ice-covered lakes. There's a picture of Chris McKay, who's going to be in a few weeks. He's one of the few people who's gone through these holes that are, literally meters deep in the ice. They have to melt through and then go down, obviously, with the dry suit. When they come up to the surface, someone else has to pour boiling water over them so you can unfreeze the hinges. I've never actually done this, but Chris has, and that's when he used to have a beard. He doesn't anymore, and I'm sure he'd be happy to tell you what it's like if you want extreme diving. If you go to the bottom of these, you see that they're microbial mats, and as you know, there are few things in life that cheer me up more than a nice microbial community. And there they are at the bottom of these ice-covered lakes. Unbelievable. Here's Nymph Creek again, which I think I already showed you this picture. What's interesting is the one over on the right. This is right near Nymph. This is Iron Spring. Boiling water. I don't know if you can see the sort of crinkly bits on the surface. So there's steam coming up, and as soon as that steam cools off enough, you see the cyanidium again. So that's the acid steam. And I promised I'd take you very briefly to Africa. We've been privileged to go there twice in the last few years. We've been doing partly field work and partly educational work with a colleague down at the Jet Propulsion Lab, Kevin Han. We've gone into some of the schools. He's really led a marvelous program called Cosmos. If anyone's interested in that, I'd be more than happy to talk to you about it later. There you can see some of the sites through the Rift Valley that we've been to. And again, you notice these pHs, Lake Bogoria. I've showed you a picture of that with the flamencos. One corner of that's pH 11.4, Lake Elementaita 10.2, Hell's Gate only 9.5. I mean, this is unbelievable. And again, it's because they're endorheic. So the only way that you can lose water from there is from evaporation. And this is what some of these sites looks like. There's Hell's Gate over on the left. Actually, there was a film that was done there. It's an Indiana Jones, something like that, through part of that. Very cool area. Hot, but cool, if you know what I mean. There's some of Lake Bogoria, again, with the flamencos, and I promised I'd point this out. You can see the footprints of the flamencos look green. It's because there are so many cyanobacteria in there. It's almost like cyanobacterial soup, that when they step down, you can see squished cyanobacterial sort of footprints there. There's Kyle out at Lake Elementaita. See, he's fine. About pH 10, still alive. Anyway, you can see what I do to my kids. Now, he actually had a great time. That was our last day in Kenya. And up above that was a site that I was really excited because there were fish in there and it was high temperature, just sort of at their limit. I thought this is going to be a great field site because of the whole, solubility of oxygen problems. And then I discovered it was also doubled up as a Maasai laundromat. So that site was sort of off, but I thought it was kind of fun showing the pictures of the Maasai who'd come out of nowhere to do their laundry there. Here's a very low pH site. I'd like to flip you back and forth. This is again Yellowstone. This is Roaring Mountain. You can see the steam vents coming. There's a group of acidophilic thermophiles that live there, and they produce sulfuric acid, so they're eroding the mountain. This is one of those organisms that's useful in mining. Here's a lake in New Zealand. You saw this picture when we talked about origin of life, because to me that is the picture of Darwin's warm little pond, where he sort of imagined life starting. I always imagine that lake. Again, there are places in New Zealand that are very similar to Yellowstone, high temperature, some low pH and so on. You see very similar sorts of organisms there. As you know, we've been spending a fair amount of time the last couple of years up in the Bolivian Altiplano. We come up through Chile into Bolivia, and what that picture shows up there is a satellite image of ultraviolet radiation on the surface. And so we find that these satellite images are extremely useful in planning vacations. You just go for where there's very high levels of UV, and you can see the sort of pink along by Bolivia up there in that satellite image. I'm sure many of you do that yourself, so I won't belabor the point. High UV, low oxygen, low precipitation, cold, windy, unusual water chemistry, you know, an extremophile heaven up there, and so much unexplored. I mentioned you go up through Chile. This is sort of the last outpost before the border with Bolivia, San Pedro. There's a little church there. They actually have some remarkably good restaurants and very fine Chilean wine, so it's hardly a burden to spend a few days acclimating. This is about 7,000 feet or so in San Pedro, so you get your fill of the good wine and you get a nice hot shower, and then it's off to Bolivia, across the border, and you can see the mountains up there as you come up, and very quickly you lose any possibility for trees or anything else like that. There's the border crossing in the winter, so remember, southern hemisphere, so we're talking about July. I think I took that when we were doing that filming, the movie I showed you last week. This is not, as you can see, one of your high-tech border crossings. There's usually a border guard there. It's not entirely clear to me how many of them can read your passport, but sometimes, just in case you don't feel like being on your best behavior and polite enough, they often have machine guns in there. So, you know, I'm always as polite as I possibly can at the border crossings. And if you're lucky, you're into Bolivia, and it's just beautiful and amazing up there. There's Laguna Blanca, which is a lake that's got some very interesting organisms in it. Again, it's just, you know, barren, no man's land sort of thing. It's a quick experiment that a colleague of mine did where they put UV blocking plexiglass over a section and UV transparent for a year, and you can see, again, the differences in pigments. Many organisms produce pigments to protect themselves. That was actually filming that show. I thought you might sort of enjoy that picture. Those shots when you're driving are not as easy to film as you might think, because they're asking you these questions, and you're supposed to be looking forward and there and driving and trying not to go over the road at the same time. And, I think the worst was, there was one, I was really tired up there. I'm driving, and it was one of the first questions. This nice British producer said, "Tell me, what do you think of God?" I'm like, what? So they're not, you know, they're not always as easy as you think. Sol de la Mañana, I think that was pointed out in that movie too, where the boiling mud pots were, and that's sort of the hellish Archean vision of origin of life. Laguna Colorada you've seen before too, high salt, you can tell immediately because of the red color. There's some close-ups there, flamencos, so you know it's an awkward location. And then that brings us to Australia. Australia is very interesting because it's got the high UV radiation, but it turns out there's also a site up in the Flinders Ranges that is near a uranium mine, and so therefore we have high levels of ionizing radiation. Anyone here ever been to Australia? Great, cool. So I've only been to this one trip so far, but working on another one. Anyway, we flew from Sydney to Adelaide, rented a car, and went up to Arkaroola. And so I'll give you the quick travelogue. You know, you start off, for those of you who've not been to Australia, and, you know, it basically looks like Kansas. Obviously not. There's some, you know, mountains in the background, but actually it doesn't look unlike Idaho. I'll say it may not look like Kansas, but it looks a little like Idaho. And then you see your first emu, and you realize that you're not actually in Idaho anymore. And, you know, there are dead kangaroos on the road instead of dead deer. And this actually was my favorite. This was a rest stop along the side of the road, and I'll show you a blow-up of the sign in a moment. There, snakes watch your children. So this is today's lesson on why punctuation matters. I mean, think about that for a moment. So we watch the snakes watching their children. And then you ultimately get near the field site. As I mentioned to my colleague, this is not actually what in the United States we would call a road. We would call this a dried stream bed. At some point, I got so rattled, I got out and took pictures of him going through there. This was not an easy drive. There's one of my little friends I saw up there. There's a little euglena, a little protist with a red eye spot, beautiful little thing. And then you get to a place like this. This is Paralana Hot Springs in the Flinders Range. And what you notice immediately is people in matching orange shirts. Now, this is not typical of Australia. This is, however, typical of Heathcote, the Beverley uranium mine, which is near there, has all sorts of people who obviously are around radiation a lot. And they have these hats so that they're protected from the Australian sun, and they seem to have this orange uniform. They came to help us out that day, Roberto Anatori, who's a colleague of mine, was over on the right. He lived in Australia at the time and has done most of the work up at that site. So they were very nice. They came over with their Geiger counters and so on. You see that hot spring in there doesn't look too bad. I mean, how bad can it be? Until you get close up and you see these bubbles that have thorium and uranium and things like that in it. So really you don't want to make lunch out of it. So that's sort of the radioactive site. So there are even natural radioactive places. Then you can go up into the air. And we've done almost nothing up there yet. But it's a very interesting place to be if you're an organism. You've got huge problems of desiccation. You've got huge problems with radiation exposure. You don't have water, obviously, you're desiccated. Trace gases are a problem. If you're an animal, there's not a whole lot to eat. You don't see a whole lot of hamburger stands up in the clouds. As many things in microbiology, Pasteur did much of the early work. As you can imagine, there's military involvement because you can think of biological warfare, germ warfare being through the air. Today, there are a lot of people interested in aerobiology because of preservation of monuments and also, of course, disease spread. There was a lot of work done by the military. Most of that work is not published. It's under lock and key. Fort Detrick, is it? And that could be all she wrote, except that Rocco, who just walked in, has done some aerobiology over the years, and I've gotten interested. And one of the things we're particularly interested in is this question of how high the biosphere goes. I'm sure there's plenty of stuff floating around in this room, but at what point does it start to drop off? There have been sporadic studies with high-altitude balloons. You see these published now and again. But that's not good enough as far as we're concerned. And so we've been involved in a project that's taking off very rapidly, both literally and figuratively, where we're going to be doing a lot of air capture work and really getting into this whole question of what's up there over the next, I hope, 5 years or so. The engineer who's doing the work is a graduate student of mechanical engineering who took this class about 3 years ago, Diana Gentry. We've had other people involved in it, particularly when we were doing a lot of the high altitude ballooning through the AeroAstro class and through this class, and periodically we've had groups from this class then meet in the spring and get involved. So that's sort of an advertisement, depending upon how things go now that we're getting involved with rocketry and not just ballooning, that there is potential, particularly for underclassmen, but even the elderly of you to get involved. So this is a very interesting area, I think, for many reasons, and has been seriously understudied. And so this was going to give you an advertisement for these balloon programs that we've been doing with the AeroAstro group. That was our first flight. You know, home movie. You have to show the home movie. And there you go. And we had experiments down at the bottom. You just about see it. That orange and yellow thing was a parachute. Is at some point it gets to an altitude that we've determined, the balloon bursts and the parachute deploys as it comes down, and we get our samples back, driving through all over Hellengone. So it's all very exciting. This is what it looks like up there. Our record so far with the balloons is 106,000 feet. These transcontinental aircraft go at about 37,000. So we're about 3 times as high as that. With the rockets, we can get 3 times as high as this. We can get up to over 300,000 feet. So this is very cool stuff. All of a sudden, we feel like we have access to what's technically called near space, but it feels like space. So I'm going to just finish. I know I'm keeping you over today, but I want to finish with a few random thoughts. Early Earth was an extreme environment in several ways because of this whole solar situation that I mentioned at the beginning of the lecture. The sun was less luminous, but we know that young suns can produce relatively more UV, and we know we didn't have this ozone shield, and so life on Earth, at least on the surface, was probably zapped. So that's going to be an issue. All these other things that we've talked about before on the early Earth would not be quite as extreme, although the impactors are going to produce a lot of heat, and so they're going to be certainly openings for thermophiles. And we mentioned in the evolution lecture that it looks like if you look at the tree of life, that the earliest organisms were thermophiles. And we don't know if it's because life arose in a very high temperature environment or it's because of this bombardment period that filtered out everything else. It acted as a bottleneck that life had to go through and then diversify again. We mentioned very briefly last time that CO2 levels have changed. CO2 is what we think kept the early Earth warm. Here's a best guess model out of Jim Castine from, I guess at this point, over 10 years ago. And to get an Earth where there was liquid water, he was estimating that, again, there were several orders of magnitude more CO2 on the early Earth than today. So you start to get to what, well, actually is an extreme environment from the point of view of a carbon-based life form, because it's great to have lots of CO2 around. All these things were happening as oxygen was rising, which was helping to provide an ozone shield for protection against UV. We've never seen UV below 200 nanometers, thank goodness, because of the CO2 blocking it. But this 200 to about 320 region, where the ozone helps block us, was only going up well after life formed. Again, you can play with this slide online. But there's a lot in here, and it starts to put together some of the story that I've been picturing with where life has been going in terms of its evolution, how it fits into a planetary context, and now throwing in the atmosphere and the sun. So where are we going from here now? Well, first thing is, as I mentioned, you probably are going to need extremophiles in space, because space is a really nasty extreme environment. And so as we go to try to terraform a place like Mars, as we try to make it more Earth-like, we are probably going to want to have organisms that can withstand very cold temperatures, as mentioned earlier. There's a fair amount of CO2 on Mars, so that's good news for the plants, but the cold temperature isn't so great, high UV isn't so great, oxidizing surface isn't so great. So the extremophiles are going to help us survive into the future. They've been extremely important in biotech. As I mentioned before, they are used for body parts. So for example, everyone in here, I assume, has heard about the polymerase chain reaction, PCR, right? It's basically xeroxing up DNA. Well, the critical enzyme in that reaction is Taq polymerase. There are now a whole bunch of other ones. This was isolated from Thermus aquaticus. Well, this was originally isolated, this organism and the enzyme, from mushroom springs, which actually looks a lot like octopus, except it looks like a mushroom. And in fact, they're about a 5-minute walk away from each other. This was one of Tom Brock's students, Hudson Freeze, who worked on these organisms, isolated them, and this enzyme for his PhD. Now, he never figured out that it could have some use. Kary Mullis did, and that's why Kary got the Nobel Prize and the billions of dollars, and the whole industry took off. But it was based on this very basic work on extremophiles from years before. So that's sort of like the poster child example of how extremophiles can make you billions and get you, a Nobel Prize and so on. Here's a table, again, you can play with later. Just a whole bunch of patents and so on from different extremophiles and some of the things they do. The one I like to mention has to do with bleaching paper. I forget which one that is. There's a thermophile, I guess it's a xylonases, that is able to operate at very high pH. And it turns out, well, most people, I used to look around the room and everyone had bleached paper. Now I look around the room, people don't have paper at all. But if you do have paper, I bet it's bleached. Well, it turns out the bleaching of paper is extremely expensive worldwide every year. It also has a lot of nasty byproducts. If you can loosen the pulp fibers just a little bit, you don't need as much bleach. So that saves you billions of dollars a year. And it turns out that an enzyme from one of these high temperature achillophiles is able to do that in industrial paper production. So again, saving billions of dollars just from this enzyme from one extremophile. Now, we're going to spend most of the rest of the semester talking about moving on into space. And so this is just to remind you that once we get beyond planet Earth, we're going to be talking with all sorts of other extreme environments, space vacuum, extremely cold temperatures. Radiation is no longer just going to be talking about UV. We're going to be talking about cosmic radiation, a lot of ionizing particles, things like alpha particles, heavy ions, protons, electrons, and so on. It is really nasty out there. You don't want to go out into space without helmets and spacecraft and so on. Follow the water. again, this is sort of an advertisement for the next couple of weeks. We'll be going to some of these places where we think it's possible that there could be life in our own solar system, following the water, but using everything we learned today as a way to inform us whether this is reasonable or an incredibly stupid idea. So, what I've talked about a lot is individual parameters like temperature, pH, and so on. I think one of the next steps from a sort of theoretical point of view is to be looking at multiple variables. You know, can you have high temperature, high salt, low pH? And if we don't on the Earth, is it because you cannot get all those combinations together? I had a friend in corn genetics years ago, and I was a grad student, and she said you could get red and white corn, but you could not get red, white, and blues. My father kept saying he'd market it for the 4th of July, and apparently you can't get all three together. It's unstable. You know, can you get all of these extreme conditions in one organism, or can you only get 2 or 3, or is it that we don't see them because the environments aren't there? And again, anyone who would like to work on that next semester, I would encourage you, because I think it would make a very cool library study to get an idea where in this multi-dimensional niche space of life we see organisms and where we don't, and why we might not see them. So let's conclude by the thought that extremophiles are intimately connected to the fact that we're based on organic carbon. That's why we have these extremes. It all has to do with keeping your body together. The story of extremophile research is very much tied in with astrobiology, although I argue that the history could actually be thought of as dealing with food preservation. The envelope for life is much greater than we could have possibly imagined, and I'm hoping, because otherwise I'll be disappointed, that it's much greater than any of you thought 2 hours ago. That you now know that there are organisms above boiling temperature of water, way below freezing, at pH 0, boiling battery acid. You know, who could possibly imagine these things? It takes the nerve to say, you know, I wonder if something's there, and be willing to act like a fool, and then finding stuff. I mean, it's unbelievable. Some of these adaptations are incredibly simple. Life is lazy. It's relatively simple to live at low pH. I don't recommend trying it tonight, but you know, you could probably engineer yourself ultimately to do that with some good proton pumps. Some of these things are extremely difficult. And I was Googling one day, I found this quote from George Bernard Shaw: "A reasonable man adapts himself to the world. The unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man." And I guess much the same can be said for the extremophiles.
For more, please visit us at stanford.edu.